Hi, my name is Rachel Marcus, and my study is the effects of radial water maze training and ketamine administration of paravalvular expression in the rat hippocampus. Schizophrenia is a prevalent disease in our society that can be aided with medications but cannot be cured. Researchers have been using schizophrenic models with the use of ketamine to have a more in-depth view of the effects of schizophrenia in the brain. Recent studies have found that in animal models, paravalvular expressions are lowered when given ketamine. Other studies have also stated that when animal models are trained, the paravalvular expressions were increased. By combining these two inferences into one study, researchers will gain crucial insights to schizophrenia. In this study, I will be looking at histological data of rats that have been treated with 30 milligrams per kilograms of ketamine that have either been trained in a radial water maze or have not been trained. Starting in the summer of 2013, I've been fortunate enough to work under the mentorship of the James J. Kropak Lab at the University of Connecticut. With the help of Dr. James J. Kropak and graduate student Jennifer A. Corvey, I was trained to handle animals in different histological methods. In July 2013, I joined the Kropak Lab with the already trained rats and gathered data for 3 weeks of the trained rats in the radial water maze. By late July 2013, I helped gather histological data with Jennifer Corvey. From August 2013 until October 2014, Jennifer Corvey had been slicing and staining different areas of the brain for the expressions of paravalvular and creating slides. In November 2014, I am analyzing the stained slices of hippocampal regions using the ImageJ software. This software is used to count specific cells needed while looking at the images. After finding the average amount of paravalvular expressions in different sections of the hippocampal region, such as CA1 and CA4, data will then be compared between the ketamine-treated trained rats and the ketamine-treated untrained rats. In a study conducted by Patricia O'Donnell at the University of Maryland School of Medicine in 2011, researchers found that human participants with schizophrenia had a dramatically reduced paravalvular expression, specifically in the CA4 hippocampal region. Another study in 2012 at the Center for Neuroscience at New York University trained adolescent rats with prenatal hippocampal lesions. After training the rats with cognitive tests, paravalvular expression stainings were collected. Data showed that the rats with the ventral hippocampal lesions and trained throughout adolescence showed an increase of paravalvular expression in the hippocampal region than the neonatal ventral hippocampal lesions rats who were not trained. These separate findings help support my research. Since ketamine is a method of enhancing schizophrenic symptoms, the trained rats in my studies should have a higher paravalvular expression than the treated untrained rats. After gathering slides from each of the 18 trained and untrained rats, I will then use the ImageJ software to analyze pictures of the slides by focusing only on the paravalvular stainings and counting the amount of protein in the given sections. After finding the amount of paravalvular in each section of each rat, I will then take the average paravalvular expression in each hippocampal region from the trained and untrained rats. After analyzing the expression data, I will then compare the averages to see if there is a difference between the expressions of trained and untrained rats that were both treated with 30 milligrams per kilograms of ketamine.